Good evening, everyone. Today we are discuss about the structure of RNA and DNA. In the previous lecture, we only studied about the structure of DNA, which is simply called deoxyribonucleic acid. If you can see the structure of DNA, that consists of a double helical shape or double helical structure. That is the two strands. One strand is here, one strand is there. That is the two strands consist of double helical structure. But in case of RNA, that is the ribonucleic acid. If it is the complete name ribonucleic acid of RNA, that consists only the single helical structure. You can see that consists only the single helical structure. The most commonly question asking in the exams, that who discovered DNA and who is the father of DNA. If that is the question is who is the father of DNA, that is James Watson. James Watson is the father of DNA. And who discovered DNA? That is Friedrich Mischer. ओके, तो फ्रेडरिक मिशर जो थे, इन्होंने क्या किया था, डिस्कवर किया था डीएनए को, डिस्कवर डीएनए. एंड द फादर ऑफ डीएनए इज जेम्स वॉटसन, फादर ऑफ डीएनए. Okay, so mostly time the asking question in the exams, that what is polynucleotide chain or nucleotide chain. न्यूक्लियोटाइड चेन और पॉलीन्यूक्लियोटाइड चेन के बीच में एक मेजर डिफरेंस ये आता है. न्यूक्लियोटाइड चेन उसको बोल देते हैं जो सिंपली एक सिंगल फॉर्म में प्रेजेंट है. पॉली का मतलब होता है बहुत सारी मोर चेन्स प्रेजेंट, दैट इज मोर न्यूक्लियोटाइड चेन्स ऑफ डीएनए एज वेल एज इट इज आरएनए. यहाँ पे सिंपली आप ग्रुप्स देख रहे हैं. यू कैन सी द 4 मेजर ग्रुप, दैट इज साइटोसिन, ग्वानिन, एडेनिन एंड थायमिन. And in case of RNA, there are also 4 groups, that is cytosine, guanine, adenine and uracil. These groups are called nitrogenous bases. Okay, if I am talking about the DNA, the 4 nitrogenous base, that is adenine, thymine, guanine and cytosine. That is in case of DNA. In case of RNA, adenine, uracil, guanine and cytosine. The major difference in these two, the major difference in their nitrogenous components, at the place of thymine, in case of RNA, uracil is present. So thymine is replaced by uracil in case of RNA. Okay, so these are the major difference of the one nitrogenous component in case of RNA, that is there is uracil is present at the place of thymine. Okay, so simply अगर मैं कहूँ component of DNA and RNA, components of DNA and RNA. Components means the different constituencies which is present inside DNA and RNA. One thing is the nitrogenous bases. One is nitrogenous bases plus phosphate group plus pentose sugar. So these are the 3 components which is present inside the genetic material, as it is RNA or DNA, nitrogenous bases, phosphate group and pentose sugar. So these are the 3, and these 3 are totally called nucleotide. Very important, what is nucleotide? That consists of 3 component, that is nitrogenous base भी हमारे पास जो कंपोनेंट प्रेजेंट कर रहा है, फॉस्फेट ग्रुप भी है, और एक शुगर प्रेजेंट है जिसका नाम है पेंटोज शुगर, जिसमें 5 रिंग हैं. इस तरह से इसका स्ट्रक्चर कुछ इस तरह से बनता है, रिंग लाइक स्ट्रक्चर, दैट कंसिस्ट ऑफ 5 ग्रुप एंड कंसिस्ट पेंटोज शुगर. इन दिस इज कॉल्ड न्यूक्लियोटाइड. समटाइम्स क्वेश्चन इज आस्किंग न्यूक्लियोसाइड क्या होता है, व्हाट इज न्यूक्लियोसाइड. In this only nitrogenous bases है प्लस शुगर है, that is pentose sugar. Only these 2 components make nucleoside. यहाँ पे phosphate group absent होता है. In this nucleoside, the phosphate group is absent. That consists of only the two component, that is the nitrogenous base and the pentose sugar, that makes a nucleoside chain. Okay. And sometimes in the exams a question is asking like that, draw the structure of polynucleotide chain, draw the structure of nucleotide chain. Then you make the first of all the phosphate group, and one is the pentose sugar and one attached of nitrogen space, that complete the nucleotide chain of DNA as well as RNA. So in case of RNA, you can see that the single helical structure is present, and these नाइट्रोजनस स्पेसिस जो है इस तरह से यहाँ पे अटैच होते हैं. जैसे मान लीजिए एडेनिन यहाँ पे है, यूरासिल, ग्वानिन एंड साइटोसिन लाइक दिस. ऑन द अदर साइड दीज आर अरेंज्ड लाइक दिस, 4 ग्रुप यहाँ पे एंड 4 ग्रुप ऑन दिस साइड. बट इन केस ऑफ DNA you know that is the double helical structure. ओके. सो जो ग्रुप है वो इस तरह से अटैच है, 4 ग्रुप लाइक दिस. यहाँ पे 2 स्ट्रैंड है DNA के प्रेजेंट, और RNA में क्या है हमारे पास सिंगल स्ट्रैंड है. एंड यहाँ पे RNA से क्वेश्चन आता है हु डिस्कवर्ड RNA, तो दैट इज रिचर्ड अल्टमैन. रिचर्ड अल्टमैन ने RNA को डिस्कवर किया था. ओके. सो सिंपली यहाँ पे जो मेजर डिफरेंस आपको मैंने पहले भी बताया, दैट इज़ यूरासिल is present at the place of thymine in case of RNA. That is only the major one difference in case of RNA. And the second difference is that there is only a single chain helical molecule in RNA, and there is a double helical structure of DNA. And question is arises from this side, ये double helical structure किसने दिया था? That is Watson and Crick. Watson and Crick gave the double helical structure or double model helical structure of DNA. These are the very important and most asking question in the exam as well as in your competitive exams.